Hello everyone. Today we will discuss about the structure of the seed. We know in the life of a flowering plant, seeds are very important structures as they help in the formation of new plants. A seed is a mature ovule after fertilization and it is covered with a protective covering called seed coat. Seed coat consists of testa and tegmen. Testa is the outer, hard, protective covering and tegmen is the inner, thin membranous covering. On the basis of endosperm, seeds are divided into two types, endospermic and non-endospermic. Endospermic seed. In endospermic seed, cotyledons are thin and membranous and endosperm persists. For example, custard apple. Non-endospermic seed. In non-endospermic seed, cotyledons store food and endosperm does not persist. For example, peas, beans, etc. Broadly, seeds are of two types, monocotyledon seeds and dicotyledon seeds. Monocotyledon seeds are the seeds in which there are only one cotyledon. For example, maize bean. Dicotyledon seed. In dicotyledon seeds, there are two cotyledons in it. For example, bean seed. The structure of the monocot seed can be studied by taking an example of a maize grain. A maize grain is actually a fruit in which the seed coat and the pericarp of the fruit wall are fused together to form a protective layer. That is why it is called as grain. If we look at the structure of a maize grain, we will find small light-colored oval-shaped area in one side. This area marks the location of the embryo inside the seed. The remaining large part is endosperm, which is rich in starch. The endosperm and the embryonic part are separated from each other by a thin epithelial layer. The outermost layer of the endosperm is rich in protein and is called Aryan layer. The embryo consists of a single cotyledon called scutellum, a radicle, and a plumule. The radicle is towards the pointed end and it is enclosed in a protective sheet called coleorhiza. The plumule is towards the upper broader side of the embryonic region and is enclosed by a protective sheath called coleoptile. The structure of a dicotyledon seed can be studied by taking an example of a bean seed. There are a number of different kinds of beans such as broad bean, lima bean, etc. But the general structure of the bean are same. Most are kidney shaped with a convex and concave side. It is covered with seed coat which contains testa and tegmen. If we look at the convex side of the bean seed, we will find a whitish oval skirt which is called hilum. Hilum marks the area where the seed was attached to the wall ovary. A tiny pore called micropyle is situated close to the hilum. Seeds absorb water through this micropyle. Below the seed coat are two thick cotyledons, which contain food for the embryo and protects it. On carefully separating the two cotyledons, the tiny embryo can easily be seen attached to one of the cotyledons. The embryo consists of two parts, the radicle, which later forms the root, and the plumule, which later forms the shoot. The region of the axis between the point of attachment of cotyledons and the plumule is called epicotyle, and the region of the axis below the cotyledons is called hypocotyle. Thank you.